Welcome back in a new video session from Biology Boost Up. Today in this video series we going to study about some basic concept regarding DNA replications as well as RNA transcription. So now let's start. At first, the concept of DNA replication. Now look, the replication is a process of formation or synthesis of DNA from a DNA itself. Second, the replication occurs at the time of S phase of interphase of cell division. Third point, the process of replication started at a particular point, that is the origin of replication or ORIC, and then it occurs or moves on both directions. The replication occurs 5' to 3' direction. Next, the replication follow the semi-conservative model. Next, each strand of parental DNA acts as a template strand. Next, the replication started at the presence of RNA primer. And the last point, the unit of DNA replication is known as replicon. Now the next topic is the proteins or enzymes involved in DNA replication. First, the helicase or DNA B protein. This enzyme helps to separating the DNA strand at the point of Y-shaped replication fork region. Next, topoisomerase 1 and 2. This enzyme helps to opening of supercoil of DNA or template strand of DNA. Next, the DNA polymerase 3. This helps to synthesis of new DNA strand over the template parental strand, and it has 3' to 5' exonuclease properties as well as proofreading activities. The ligase. This enzyme helps to joining of fragments of DNA strand. Next, DNA polymerase 1. It helps to denaturation of RNA primer and has a proofreading activity. Next, the single strand binding protein or SSBPS. This protein joined with each strand of DNA at the fork region and prevent to bond formation between two separated strands during replication. Next, the DNA A and DNA C protein. The DNA A at first helps to unwinding of DNA at the fork region of DNA. Now look at the some basic concept regarding transcription or RNA synthesis from DNA. First, the initiation site. The DNA sequence to which RNA polymerase binds to initiate transcription of a gene is called the promoter. They are called the -10 and -35 element, denoting their position relative to the transcription site, which is defined as the +1 position. Next, the upstream. It is the direction 5' from which RNA polymerase has come, that is the sequence preceding the start site left side of the 0 point is designated by negative value. Now look at the downstream sequence is the direction of 3' of transcription, that is right side of the 0 point and designated as +1. Next, the Pribnow box or Tata box, a DNA region where RNA polymerase binds to initiate transcription, that is the regulatory site of DNA which have two conserved or unchanged sequence -35 region and -10 base upstream from initiation site, which has the 5' Tata 3' in prokaryotes is called the Pribnow box, and the -35 sequence is known as recognition site. Thanks for watching my video. Please like, share and subscribe my channel. Thank you friends.